All right. Make sure I'm recording the audio as well. Pretty sure that I am. All right, so here's the paper. Illuminating the Evolutionary History of Chlamydia. This is also a genome sequencing project. But when you read the paper, you might not realize that they did all that work. All the sequencing data is in the supplementary information. So this is about an organism called UWE25. It's since been named Protochlamydia amoebophila. This is what they call an environmental chlamydia. It is not a free-living organism. It is also an obligate parasite, just like the chlamydia that you're used to thinking about. It has reticulate bodies that live inside the cells of other organisms, and elementary bodies that are the infective particles, or basically a kind of a spore that floats around, in this case, in the environment rather than in interstitial fluid to infect new cells. This thing infects amoebas. Environmental chlamydia are everywhere, and they form a unique phylogenetic group. And so here's a tree of right here that shows the pathogenic chlamydia. These are what they're calling them pathogenic. These are the pathogens of plants and animals. And then the environmental chlamydia. They're able to sequence the genome of this thing. Although you can't grow it in pure culture, you can grow amoebas, infect them with chlamydia, and then physically separate out the elementary bodies from the culture media. And then isolate DNA and sequence the genome. These organisms are interesting because although they are obligate parasites, they are not as far down the road of reductive evolution as the plant and animal pathogens are. And the reason for this is unclear. But they start right off by saying that the genome of this thing is about twice the size as the genome of Chlamydia psittaci or Chlamydia pneumonia or Chlamydiophila, etc. They also show that this genome is stable. There aren't a bunch of pseudogenes in it. There aren't a bunch of transposons and that kind of stuff. This genome seems to have stabilized at its current size and state, and is not continuing the road down reductive evolution the way other pathogenic chlamydia have. They use this as a kind of missing link. And so they're thinking about, parasitic bacteria and obligate intracellular parasites like chlamydia, and then they think of this organism as partway but not all the way down that path. And if you read the paper, you can see they're very cognizant of the fact that this is dangerous. That this organism, UWE25, is not the ancestor of chlamydia. It is, that they hope, a bit like the ancestor of chlamydia. But it's a modern organism, alive today, stable as a parasite of amoebas. They argue that the pathogenic, and they use the terminology pathogenic for the plant and animal pathogens, and environmental for the ones that infect amoebas and other protists. They say that pathogenic amoebas thrive in a more homeostatic niche. What does this mean? Chlamydia, the pathogenic chlamydia, doesn't have to float around in rivers and streams or sit in the dirt before it can infect another organism. Pathogenic chlamydia require direct contact of bodily fluids. And so they're never outside, in the cold. They're never exposed to salt or freshwater environments. They're in basically a body environment the whole time, whether that's from a plant or an animal. And most of the ones that have been studied are in animals. And they argue, although it's not clear to me why it would be, that results in them not losing as many genes and being more flexible about different conditions than the pathogenic ones. But bear in mind that those elementary bodies are metabolically inert and just float around in the environment. They just have to survive, is all. And one of the ways that they point that out is that this UWE25 has more ribosomal operons than chlamydia do. They've got some interesting arguments here. They look at the genes, and they see a significant number, as is seen in chlamydia proper, the pathogenic chlamydia, of genes that are most similar to genes in cyanobacteria or chloroplasts. And so they argue that chlamydia and cyanobacteria are probably specifically related as phyla. This is probably true. But it's also true that a lot of pathogenicity islands look like cyanobacterial genes. If you look at pathogenicity islands in Listeria, you'll find cyanobacterial genes in there. And so that may be an issue of pathogenicity islands more than it is a specific phylogenetic relationship in those groups.
Are there a lot of pathogenic? Are there any cyanobacteria that are pathogenic?
No, they're all phototrophic autotrophs, right?
Okay.
However, there are many of them that form symbioses with plants and animals. And that tight association is like a pathogenesis, and it's those kinds of association genes that are the ones you see in pathogenicity islands. It's not the cyanobacteria are also experts at making secondary metabolites, many of which are pretty awful. Toxins, things like ciguatoxin that causes ciguatera, hallucinogenic alkaloids, and you know, all this kind of stuff. Oh, you'd be amazed. Most of the toxins, well, red tide toxin, for example, is the result of the growth of a dinoflagellate. That dinoflagellate doesn't make the toxin. It's the cyanobacterium inside it that makes the toxin, and that's quite common. Let's see. It's an obligate intracellular bacterium, like all of them, and it has reduced metabolic capabilities like other chlamydia. But it does have a complete TCA cycle. And it needs this to make a bunch of amino acids that the pathogenic chlamydia don't make. They make the argument later on here is that they, well, no, right here, that they're oxytropic. They require in their diet, if you will, most amino acids. But the reality is UWE25 makes a lot more amino acids than the pathogenic chlamydia does. And although they don't mention it in this paper, they mention it elsewhere. A really interesting one of these amino acids is tryptophan. In pathogenic chlamydia, the ability to synthesize tryptophan is a virulence factor. And that's about the only amino acid that pathogenic chlamydia can make. But they can make tryptophan. And if you knock out the genes for tryptophan biosynthesis, those strains are avirulent. And so somehow the host organism, presumably, is reducing the parasite's ability to get tryptophan. It doesn't get enough tryptophan from the host. It has to make its own. Probably not completely eliminating the parasite's ability to get tryptophan, but reducing it enough that it causes harm to the parasite, slows it down, and that gives it just that little extra edge that allows the host to take over, to get that edge on it. UWE25 can't make tryptophan. It's one of the amino acids it can't make. And it turns out that there's pretty good evidence that this organism functions more as a symbiont than a parasite. It rarely lyses the amoebas. Usually it grows in the amoebas in a vacuole, and those vacuoles are the contents of those vacuoles are expelled, and the amoeba survives. Both pathogenic chlamydia and UWE25 have an abbreviated respiratory chain. E. coli has a very complicated respiratory chain, but when grown under microaerophilic conditions, it only uses part of it. And that respiratory chain, maybe can generate proton gradient in UWE25, unlike chlamydia, where it can't. It's absolutely clear that UWE25 has an ATP-ADP antiport. So it's sucking ATP directly out of the cytoplasm of its host. It is an energy parasite, just like pathogenic chlamydia are. And as a result, it has no need to make nucleotides or any of that kind of stuff because it can get them straight from the host. This organism has two secretion systems. It has a type 3 secretion system. They call this TTSS. I don't know why, but they do. And UWE25 has a type 3 secretion system. What's interesting, later on in the paper, they tell you what this system does in pathogenic chlamydia. In pathogenic chlamydia, this type 3 secretion system seems to be used to inject this CPAF, protease-like activator factor, into the host cell. This protein, either directly or more likely indirectly, causes the turnover of human or host transcription factors. This is a way the parasite uses to take over the metabolism of the cell. And particularly of genes for the major histocompatibility locus. Now, what good would that do? Remember, this is in a multicellular creature in these parasitic or the pathogenic chlamydia. And macrophages are constantly touching all the cells in your body to look for infected cells so that it can kill them. And it does this through the major histocompatibility locus. And so by turning down the MHC complexes on those infected cells, it reduces your immune system's ability to recognize them and to destroy them. And so that's a virulence factor in the pathogenic chlamydia. The environmental chlamydia, it's certainly, that type 3 secretion system is clearly there, but it clearly is not injecting transcription factor turnover proteins. Amoebas don't have major histocompatibility genes. So it's doing something different in those organisms, and what that is unclear. They also have a type 4 secretion system. In pathogenic chlamydia, this is used to inject other proteins into the host cell, but what those proteins are and what they do is entirely unclear. And so here we have the genome of a chlamydia that has the same life cycle as the ones that infect humans, but it has gone through much, much less reductive evolution. And by comparing the genomes of these two organisms, you're able to learn a lot about not just how this environmental amoeba-infecting chlamydia works, but it also tells you a lot about how the chlamydia you already know a lot about work. That's comparative genomics. The way I like to think about this, you know that in an experiment you have to use controls, right? Many students think of a control as a way to alert you to a problem in your experiment, right? You know, if the band doesn't show up, then your PCR shouldn't work or something like that. That is not the purpose of a control. Every measurement in a scientific experiment is a comparison between two things. The controls are what you're comparing it to. And so when you make a change in an amino acid of a protein and measure the, the kcat of that protein, the catalytic rate, that number is meaningless by itself. You have to compare it to the protein without the substitution or with a different substitution. It's those comparisons that matter. And that's how genomes work too. you learn about a genome by looking at the phenotypes of the organisms you're comparing and the genotypes and seeing if you can make sense of the differences in their genomes and the differences in their phenotypes. That's the power of comparative genomics. All right, and so next time we're going to talk about metagenomics, looking at the genetic composition of an entire environment.